The next topic is carrier-mediated transport. In this case, we're going to use a carrier. Carrier are proteins. Proteins are located in your cell membrane. Therefore, all these different processes are going to have the presence of a cell membrane. The carrier is going to show two very important characteristics, which is going to be specificity and saturation. Specificity meaning that it's going to carry only one thing. For example, the carrier for insulin is only for insulin. It's not going to be used by any other molecule. And saturation means that if the carrier can transport only two insulins, then if you have five, it doesn't matter. It will transfer only two because it gets saturated. Okay? So when the carrier is saturated, it's going to give you the concept of transport maximum. So let's keep scrolling down and let's see what we have next. So right here, we scroll down and we have that there are three different kinds of carriers. As you can see right here, they're going to be uniports, symports, and antiports right here. Uniport transports only one substance across the membrane. Symport transports two different ones, but they both are transported in the same direction, and therefore that process is going to be called transport. Antiport also transports two things, but these two things are going to be transported into different directions, meaning one outside the cell and the other one inside the cell. That process is going to be called a counter transport. The processes that are going to be achieved by these different type of carriers are going to be three, which are going to be right here: facilitated diffusion, primary active transport, and secondary active transport. Okay, so now we have then facilitated diffusion. In facilitated diffusion, the substances go, as you can see here, from high to low. But it doesn't do it alone. It needs a transporter in order to achieve that. So then the molecules go from high to low, but with the help of this transporter. For that reason, this is going to be called facilitated diffusion. Okay, now the primary active transport, right here, primary active transport, this is done from low to high. It's kind of like going from the base of the mountain to the top of a mountain. It's against the gradient. And because of that, it's going to use ATP. So primary active transport is an active transportation because it uses ATP, but in this case, it goes against the concentration gradient. Remember, we always say from high to low. Well, in a lot of circumstances, it's going to be done from low to high, but that is only if we are going to use ATP. In other words, if we use energy to achieve that purpose, which is moving things from low to high levels. Okay. Secondary active transport, in this case, is we use two transporters. Now, let me see the figure in here. Right here, you can see have one transporter, another transporter. This transporter, you have it in here high, goes from high to low. Now inside the cell is going to be high because you transported all these things inside. Now from inside it's high. You're going to move it outside where it's low through this other transporter. So it's called secondary transporter because it has one and two transporters. Okay. Very important is the sodium potassium pump. And that's what you see right there. Sodium potassium pump is a very important pump. It's a very important transporter. It is a very important transporter because this sodium potassium pump will allow us to maintain something very important, which is right here, maintenance of the membrane potential. See here, sodium potassium pump. You're going to see this several times in the other chapters coming up. Okay, so it's very important for us to understand that the sodium potassium pump maintains the membrane potential. You're going to see that later on. In addition to maintaining the membrane potential, obviously the sodium potassium pumps has other functions such as regulation of cell volume, heat production, etc. But the one that you need to remember is maintenance of the membrane potential, and we're going to see later on. Now, after that, we have vesicular transport. As you can see here, you see these are vesicles and they get transported inside. Okay, vesicular transport is used for the transportation through the membrane of large amounts of substances. In the case of this type of transportation, you're going to have two very important concepts. One of them is going to be endocytosis, when you put things inside the cell, like in this case, you see they are outside, and they are being sent inside. Now, eventually, these molecules are going to be inside. So this is endocytosis. The other concept is going to be exocytosis, where you look at this backwards. So they are inside, the vesicle opens, the substances get outside. Okay? So in vesicular transport, we have two very important concepts. One of them is going to be endocytosis, and the other one is going to be exocytosis. Examples of endocytosis are going to be receptor-mediated endocytosis, which are these ones are the receptors. You see, the molecules are going to use these receptors in order to get the vesicle, and then they're going to end up inside. Another one is pinocytosis, where these molecules right here, they are going to be extracellular fluid. So pine is basically for, let's say, water. Right? It's for liquids, for fluid. So that's why the pinocytosis means that water moved inside the cell using these vesicles. Another important example is going to be phagocytosis. It's another example of endocytosis where the cells, especially those cells that are going to defend our body, are going to get these, let's say, brown circles. Let's say they are bacteria. This is the cell that defends our body. And the way that cell that defends our body is going to capture these bacteria in order to destroy them is going to be by the way of endocytosis. So let's say these are bacteria. Then our cell is going to form a vesicle like this, and then the bacteria ends up inside that cell that eventually is going to destroy this bacteria. So if we scroll down, we're going to see here another concept, which is transcytosis, which is basically the transport of molecules from one side, from here, one side, and then they are delivered to the other side. Okay, that's transcytosis, from one side of the cell to the other side of the cell. So if we scroll down, now we have in here an example of exocytosis, which I already mentioned, right? We said this is the vesicle. The vesicle gets in contact with the cell membrane, and the contents get released to the outside. That's going to be obviously exocytosis. If we keep scrolling down, we're going to see here that there is a table showing you all the different types of transportation that we have mentioned so far in this chapter, and that's about it. Okay, the next topic is the cell interior. If we scroll down, we're going to see that the first thing we have in there is the cytoskeleton. The skeleton is going to give the cell the shape that it has. Remember, we said that we have cells that look like cubes, columns, like stars, right? Because the cytoskeleton is these structures right here that are going to give the cell the shape that they have. Okay, what are these cytoskeleton made of? Then they're going to be composed of structures called microfilaments, as you can see here, microfilaments, intermediate filaments, and microtubules. They're basically like bones that they're going to allow the cell to maintain and to keep the shape that it has. Okay, so they are made of proteins. In addition to that, what else we have inside the cell? Then we have cell organelles, right? So this is the cell, for example, right? And you have in here the nucleus, right? The nucleus is where the genetic material is located, such as the DNA and the RNA. Right here you have other structures that you have. You can see here, this is the Golgi apparatus, the rough endoplasmic reticulum. These are going to be ribosomes, right? And you just keep scrolling down. And we can see endoplasmic reticulum. You can have mitochondria, ribosomes, Golgi apparatus. Right here it says that the ribosomes are the area where the proteins, as you can see here, you see proteins, where the proteins are going to be created. Okay, so you can look at that, but if we scroll down, we can see a table where we have a summary of all these structures that you have right here. So right there, you can see all the structures that you have inside the cell membrane. We talk about the microtubules, we talk about the nucleus, the rough endoplasmic reticulum, right, which are like tubes that have ribosomes on top of them, and they are important because they are going to be related to the protein and transportation, synthesis and transportation of proteins. We have a smooth endoplasmic reticulum that are going to be related to lipid synthesis as well as detoxification. Ribosomes, we said that's the area where they are going to produce the proteins. Golgi complex, they modify the structures that the proteins are going to have in order to release them. Okay. Lysosomes, that's where as I mentioned, for example, bacteria get destroyed. Peroxisomes, they are similar to lysosomes, but what they're going to have is they're going to have chemicals to detoxify the cell. Okay, so you can look at these structures and you can see the different functions that they are going to have. We have centrioles, right? Centrosomes, and mitochondria, where we are going to produce ATP. Okay, the centrioles are going to produce the mitotic spindle necessary for the cell division, and that's about it for this part.